Let's start with our topic, and our today's topic of infectious medicine is rabies infection. The rabies is the infection caused by the rabies virus, and this rabies infection is the virus enters the body with the bite of the mammal, and these mammals can be the dog, foxes, raccoons, the skunks, and the bat. So all these mammals, when they bite the human, they inoculate or they inject the rabies virus into the body. And then the rabies infection is mainly infection that affects the brain and the spinal cord. So once the virus enters the body, there is retrograde movement of the virus in the nerves, and then it reaches the brain and cause the different complications or dysfunctions by the neuronal damage. So rabies is the infection caused by the virus, and this virus enters the body through the bite of the mammal. So dog is a very common animal that can cause the rabies infection, especially the rabid dog, the dogs which are not vaccinated, so stray dogs that moves or those lives on the streets, they are not the pet dogs. When they bite, usually they can cause the rabies infection. And the domestic or the pet, usually dogs, they get vaccinated on regular basis. So these are not the dogs that can cause rabies, but the pets or the dogs which are not kept by some family. So bite of the dog causes the wound, and the rabies virus is present in the saliva of the animal, and when that saliva touches the laceration or the abrasion, it can cause the infection. So you can see with the bite, this is the deep wound caused by the bite of the dog. And then from this wound, the infection or the virus, after multiplication in the skeletal muscle, it goes through the retrograde axonal pathway or transfer to the brain, and it causes damage to the neurons and cause neuronal dysfunctions. And two types of rabies infection is very common. Since the brain is the main organ of involvement, encephalitic type of rabies is common. 80% of the patient present with encephalitic rabies, and only about 20% will be causing the paralytic type of infection. So the rabies is caused by the rabies virus that is present in the saliva of the mammals, and different mammals like dogs, raccoons, skunks, fox bite cause the rabies infection. The etiological agent, what are the agent or what are the viruses that cause the rabies infection, is known as the rhabdovirus. So rhabdovirus is the causative agent of the rabies. Two common rhabdoviruses. We will go over some next later on, some less common rhabdoviruses also, but the more common are the lyssavirus and vesiculovirus. This is the structure of the rhabdovirus, and you can see we have the envelope. This is the envelope or the membrane. This is the matrix protein, 3 proteins you will see. Then we have the glycoprotein, that's the glycoprotein, and then the ribonucleoprotein. The rhabdovirus is the single-stranded RNA virus, and this single-stranded RNA virus has the envelope or the membrane, and then the protein, matrix protein, this is the matrix protein, then we have the glycoprotein, and then the ribonucleoprotein. So rhabdovirus is the causative agent. We have lyssavirus, vesiculovirus, and there is the envelope membrane. We have matrix protein, glycoprotein, and ribonucleoprotein. The epidemiology of the rabies. Rabies is present worldwide. Some areas have more cases reported than the other, but it is present worldwide. And the Asia and the Africa are the regions of the world which are most commonly affected by the rabies. And this is the map of the showing different regions of the America or different areas of the America where the rabies is caused by different mammals. So in this area, raccoon is the causative agent. Then we have big area caused by the skunk bite. Also here the skunk bite. Then fox is the common and mammal, and then also in this area the fox is the causative mammal. So in this American region we have raccoon, skunk, fox, and these are the mammals which cause the rabies mainly. And it's a common infection worldwide, causing roughly about 56,000 cases, and 56,000 deaths occur annually from this rabies infection. The pathogenesis of the rabies, how the rabies develop. First, as I mentioned, it is caused by the bite of the mammal. So with the bite, the virus is inoculated into the body, into the muscle. It's very important that we should, after the bite, to prevent, sometimes it's mentioned that even the good management, cleaning of the wound with the warm soapy water and detergent is very helpful in removing the virus. So after the virus is inoculated with the bite, the cleaning of the wound is very, very important. And then how this virus reaches to the brain, and from the brain to the different structures through the nerve. So after the virus inoculation, these are the skeletal muscles, and the skeletal muscles are attached to the skeleton and the bone. That's the structure of the skeletal muscle. And this replication of the virus after it is inoculated in the body, it occurs in the muscle. In the skeletal muscle it is replicated, and then in the skeletal muscle we have the junction, which is the neuromuscular junction. This is the junction between the muscle and the neuron. Neuron is the nerve cell. And in the neuron, we have the neuromuscular junction, which is the junction between the muscle and the nerve fiber or the neurons. There are the receptors are present, and these receptors are known as acetylcholine receptor. So with the bite of the animal or the mammal, the virus which is present in the saliva, it is inoculated or injected into the body with the bite, and in the muscle the virus is replicated in the muscle, and in this after the replication, it binds to, we have nicotinic acetylcholine receptors at the neuromuscular junction. It binds to these nicotinic acetylcholine receptors, and then further transmission occurs to the brain. So first step is, at this stage, we should need to, very important that we should clean the wound. We should pay attention to the wound with the warm, luke, soapy water, and that is very helpful in removing the virus from the area. And then there is replication of the virus in the skeletal muscle, and then after that the virus binds to the nicotinic acetylcholine receptor at the neuromuscular junction. Next, what happens, then the virus travels within axon. These are the axons. Axons, the neuron is the nerve cell which has the nucleus, axon, and the dendroids. Dendroids are the branches that come out of the neuron or the nerve cell. And then we have single axon. So this virus travels within axon in peripheral nerves via retrograde fast axonal transport. Now we call it retrograde axonal transport because it is going in the retrograde backward direction. The virus is inoculated in the leg, and then it goes in the retrograde transport, and it will reach to the brain tissue or the brain parenchyma. So it travels in the retrograde fast axonal transport. Then there is replication. Now first there was replication in the skeletal muscle. Next there is replication in the motor neuron of spinal cord and local dorsal root ganglia. So here the second replication of the virus occurs in the dorsal root ganglia and the motor neuron of the spinal cord. And then there is rapid ascent to the brain. Here it reaches to the brain. Ascent means going upward, descent to the downwards. So there is rapid ascent of the virus after replication in the motor ganglia and in the motor neuron and the dorsal root ganglia. It is replicated again, and then from there in the spinal cord it reaches the brain, and there is rapid ascent to the brain. After that we have, you can see here, there is centrifugal spread. From the brain there is centrifugal spread to the salivary glands, because there is excessive salivation in the rabies infection also, because the salivary glands are also involved, and there is increased salivation and drooling from the mouth of the patient who have the rabies infection. So centrifugal spread occurs to the eyes, salivary gland, to the cornea, and also to the other organs of the body. And there is also infection. Once it reaches the brain, it affects the neurons and cause neuronal dysfunctions that later on will cause early complications as a result of neurological involvement, or the late complications that involves the different organs including heart, lungs, kidneys, and cause multiple organ failure at the end. And then there is also sensory nerves to the skin. So it's the infection caused by the mammal bite. The virus enters or inoculated into the wound. From there it's goes by the nerves through the retrograde transport and ascend to the brain, cause damage of the neurons in the brain with neuronal dysfunction, and there is also centrifugal spread along the nerves to salivary glands, skin, cornea and other organs, and there is also sensory nerves to the skin also goes to the skin. Then the pathology of the rabies. This is the classical picture of the patient with the infection or with the rabies, and it is there is drooling from the mouth, and there is very, very sick, toxic appearance of the patient. These patients are aerophobic, hydrophobic also. So that's the picture of the patient with the rabies. And this is the pathological appearance. This is the neuron, and then we have the presence of virus and negri bodies in the nerve cell or the neuron, which is pathognomonic of the rabies infection. Now, what are the clinical manifestations of the rabies? We have 3 stages or 3 phases of the clinical manifestations of the rabies infection or rabies. We have prodromal phase of manifestation. Then we have acute neurologic phase, and then we have comatose phase. So prodromal stage, then acute neurologic stage, and then the comatose phase. These are the 3 phases or stages of the manifestations of the rabies infection. First, the prodromal phase or the stage. Prodromal stage, patient has the fever and there is malaise. So fever, malaise in the prodromal stage, headaches, nausea, vomiting, anxiety or agitation, and then the paresthesias. Paresthesias are also present. So all these are the characteristics of the prodromal stage. There is nausea, vomiting, anxiety, agitation, headache, paresthesias, fever, malaise. And then there is pain or pruritus. So all these are the characteristics or presentation of the prodromal stage. Next, the clinical stages of rabies. There is incubation period. Incubation period is the period from the infection to the onset of the symptom. So first, it's between 20 to 90 days, usually no symptoms in the incubation period. So it's the period from the bite to the onset of the symptoms. So in this period, it's a long period, 20 to 90 days, and no symptoms are present. Then we have prodrome, and then we know now what are the manifestations of the prodromal phase. There is after the bite, if the patient has after 20 to 90 days, there is fever, malaise, headache, nausea, vomiting, agitation, all these features are present, then there is chance that the patient have rabies developing. So prodrome is between 2 to 10 days. Next, acute neurologic disease. Acute neurologic disease and encephalitic is 80% and paralytic 20%. So 80% of the cases are encephalitic and then 20% are paralytic. And then coma or death is 0 to 14 days. So all these are the stages: incubation period, prodromal stage, neurologic stage, and the stage of coma and death. Next, the encephalitic rabies. What are the manifestations of encephalitic rabies? We know that the primary target of the rabies virus, it enters the brain and it attacks the neurons, cause neuronal dysfunction. So encephalitic, cephal is the brain. So infection of the brain tissue or encephalitic rabies, which is 80% of the patient present with encephalitic rabies. The clinical manifestations, we have initial symptoms, fever, confusion, hallucinations are patient is have visual hallucination or auditory hallucination. They see things, they hear things which are not real, so they have hallucinations. They have combativeness and seizures. So all these are the initial symptoms of the patient with encephalitic rabies, which is 80% of the patient have encephalitic rabies. They have fever, confusion, hallucination, combativeness, and seizures. Next, what are the autonomic dysfunctions that occur as a result of the rabies? Hypersalivation, increased saliva production. Patients, there is drooling of the saliva from the mouth. There is hypersalivation, goose flesh, cardiac arrhythmia, priapism, and there is hyperexcitability. All these are the autonomic dysfunction of the patient with the rabies: hypersalivation, goosebumps, cardiac arrhythmia, priapism, and hyperexcitability. Complete lucidity. Then some distinguishing features: we have early brainstem involvement. Hydrophobia. Hydrophobia is very characteristic of the patient with rabies. When they go near the water, they have fear of water, they run, they shout, whatever is their reaction, but they have fear of water. Phobia is fear, hydro is water. Aerophobia, aero is for the air, so they have the phobia of that also. Next, late complications of encephalitic rabies, because the brain parenchyma, neurons are affected, neuronal dysfunction is present. So initial early stages are not as serious as the late complications of encephalitic rabies. So some late complications are cardiac and respiratory failures. That's a serious complication, late complication of encephalitic rabies, heart and respiratory failure. Disturbance of water balance. They affect the kidneys and there is disturbance of the water balance. Non-cardiogenic pulmonary edema. Pulmonary edema is fluid in the lungs that is mainly due to the heart failure, secondary to heart failure, and this is the non-cardiogenic. So this is not associated with the heart problem, but it is non-cardiogenic and there is pulmonary edema. Gastrointestinal hemorrhage, bleeding in the gastrointestinal tract, cardiac arrhythmias, multiple organ failure. So if you see, rabies has very, very bad late complications that can occur. So it can affect the heart, it affects the lungs, it affects the fluid balance. So it affects the kidneys also. And then at the end, because of all these compromises, circulatory compromise, respiratory problem, renal problems, ultimately cause the end organ, multiple organ failure. Imaging, what findings we will have on the imaging of encephalitic rabies. Here you can see the this is the imaging of and these are all the hyper areas, increased shadowing areas which are affected by the rabies. In this imaging, then we have the thalami which are affected, and then another scan showing the hyper lightened areas. Another type, which is only 20%, I mentioned that 80% is encephalitic rabies, because the main involvement in the rabies is of the brain tissue, brain neurons are damaged. The other type, which is only 20% of the rabies, is the paralytic rabies. What happens in paralytic rabies? Paralysis, weakness is there, so it causes flaccid muscle weakness. There is flaccidity, no tone. Muscles are weak and they are flaccid. They have no tone. So if you let this hand go, it just go and lie down there because the tone is absent. So there is flaccid muscle weakness. And quadriparesis and facial weakness. Quadriparesis, all the 4 limbs are affected. Quad is 4, so paresis or weakness of all the 4 limbs, or there is weakness of the facial muscles. Sphincter involvement. Sphincter involvement is also present. Person has no control on the urine output, on the fecal output. So the sphincters are the areas or the openings which allow the food or the substance to enter from one structure to another. So if there is no control on the sphincter, they are just relaxed. It's easily cross from one structure to another. So this is sphincter involvement is there also. So there is incontinence of urine and there is fecal incontinence present. Sensory involvement is also present. The diagnosis, what are some lab investigations of the rabies? CSF, cerebrospinal fluid examination shows pleocytosis, increase in the number of the cells. Cerebrospinal fluid is the fluid that flows in the brain and the spinal cord, and since rabies is affecting the brain and the spinal cord, so examination of cerebrospinal fluid is important, and this cerebrospinal fluid is obtained by the lumbar puncture, and which the needle is inserted in the lumbar region between L3 and L4, and the fluid is taken out and is examined, and on examination it shows the pleocytosis. You can see there is increase in the cell. Then we can also perform MRI brain scans. Here, hyper-illuminated regions are present which are affected by the rabies. Then electroencephalogram. Electroencephalogram is the electrical recording of the brain activity, and it shows what are the dysfunctions or neuronal dysfunctions caused by the rabies infection. Rabies virus specific antibodies, if they are present, they are specific for the rabies infection. Then we can also, another is polymerase chain reaction amplification test, RT, reverse transcriptase polymerase chain reaction. That's another lab investigation. And then we have direct fluorescent antibody testing. So all these are different lab tests performed for the diagnosis of the rabies infection: CSF examination, MRI, antibody testing, polymerase chain reaction, electroencephalogram. Next, what are the differential diagnoses? What are some conditions that needs to be differentiated from the rabies infection? Anti-NMDA encephalitis, post-infectious encephalomyelitis, psychiatric disorder, rabies hysteria, and Guillain-Barré syndrome. So again, these are some conditions that needs to be differentiated. All these can affect the nervous system one way or the other. They need to be differentiated, and then we do the different tests like antibody testing, polymerase chain reaction, and all these different tests. Imaging techniques help to differentiate between the rabies and this anti-NMD encephalitis. This is post-infectious encephalomyelitis, psychiatric disorder, rabies hysteria. Sometimes people, even by they are so hysteric that even the little touching or scratching, they feel that they might get rabies, and they start developing the symptoms which might be caused by the rabies infection. The treatment of the rabies infection, the treatment is, we have rabies immunoglobulin is very important. We need to give the rabies immunoglobulin after the bite. And then also the rabies vaccination is also available, and this rabies vaccine is given at day 0, 3, 7, and 14 days. So it's a series of 4 vaccinations, and it's very important that this should be given. Even if there are, as a prophylactic means to prevent the rabies after the dog bite or any mammal bite. If the bite is deep enough, or there are, it's better to give the vaccination as a preventive or prophylactic measure to prevent the rabies infection. The other important treatment is cleaning the wound. Cleaning the wound with the warm luke water, deep cleaning, deep washing with soap, with detergent, and then application of some antiseptic solution or lotion is also very, very important. It says that even good soap water cleaning of the wound is helpful in eliminating the virus in some cases. So rabies immunoglobulin, vaccination, and cleaning the wound is important. Prevention of the rabies, again, as I mentioned, we have post-exposure prevention or prophylaxis, or pre-exposure prevention or prophylaxis. So post-exposure prophylaxis. These questions are important in post-exposure prophylaxis. Did animal bite patient, or did saliva contaminate a scratch, abrasion, open wound, or mucous membrane? So it's important that we should know that the saliva, because the virus is present in the saliva of the animal. So if did the animal bite the patient, or if the saliva actually touches the abrasion or the lesion. If yes, it did, Then, is rabies known or suspected to be present in the species and geographic area? So if the saliva touches or if the animal bite, then we need to know if yes, then whether the rabies is common in that geographic area or if it is suspected to be present in the rabies virus in the animal that bit. If no, then we don't need any prophylaxis for the rabies. Next, again, if it's not common in that area or if there is it's not known to be present in the species, then we don't need any treatment. But if yes, then was the animal captured? Captured, it's important to capture the animal. If no, then we should give rabies immune globulin and vaccine in suspected cases. If yes, was animal a normally behaving dog, cat or ferret? Yes, does the animal become ill under observation over next 10 days? No, does lab examination of brain by fluorescent antibody staining confirm rabies? So it's important if I just summarize this whole thing, it's important that after the bite of the animal, if the animal is captured and if the animal looks fine, we don't need any special measures. But if the animal is not captured, the condition is common in that area, we need to give the rabies immune globulin and we should vaccinate the person, and then we should look the animal if the animal behave normally or not in the next 10 days. If the animal become ill under observation over next days, then we should not give any. If yes, then we should go for the treatment. And then the treatment is rabies immune globulin and the vaccine. That's the treatment for the rabies infection. Next, we have pre-exposure rabies vaccination. That's also important before the exposure to the rabies. First, we have the 3 doses of vaccine. Day 0, 7, 21 or 28. This is the pre-exposure rabies vaccine. Make sure the post-exposure rabies vaccine, we have 4 doses, 0, 3, 7 and 14. But pre-exposure we have 3 doses, 0, 7, 21 or 28 days. Serum neutralizing antibody test. Serum neutralizing antibody test, that after the vaccination, if we look for the antibody level, if it's good enough to prevent the infection, that's the serum neutralizing antibody test, and then we give the booster doses at day 0 and 3. Some other rhabdoviruses, we have European bat, lyssaviruses 1 and 2, Irkut virus, Australian bat lyssavirus. So we have European bat lyssavirus, Australian bat lyssavirus. Then we have Duven-Haig virus, vesicular stomatitis virus. These are the domestic animals. We have all these domestic animals. These are the wild animals. These are the embryonated egg. So all these animals, we have the embryonated eggs. And then the mechanical transmission can occur just by the touching, eating, all these things can. And then we have potential non-proven. This is just the biological transfer, and then we have the biological transfer. Biological is through the environment, and then we have mechanical transfer by actually touching and consuming. So the embryonated eggs are produced, which if they are taken even by the domestic animals or the wild animals, they are capable of spreading the infection and spreading the virus. Transovarial also through the ova. Co-feeding. Co-feeding is one of the biological type that cause the infection. Contact and fomites. These animals, domestic animals, we have horse, cows, and the pigs. The contact with these animals and the fomites cause infection of the humans. Similarly, these infections by the insects, different insects, they are also transferred to the domestic animals that ultimately infect the humans. And then we have also these infect the wild animals also, and they become the source of infection. So that concludes our topic of rabies infection. Thanks for watching sqadia.com.